Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we'll be talking about annotating taxonomy from shotgun data. Now, we already discussed how the prices are different between shotgun and amplicon sequencing. And of course, shotgun gives you higher fidelity estimates of the functional repertoire, but it also gives you higher fidelity estimates of the taxonomy or taxonomic composition. So 16S is great at the genus level. Shotgun data can get down to species and sometimes even strain level. So at the beginning of the course, we discussed these two different ways of sequencing the microbiome. The shotgun approach, expensive, and if it's a completely unknown community, it's very hard to figure out which read or which sequence came from which source genome. So as an alternative, people have been using, so it's sort of the analogy here is sort of like mixing together a bunch of jigsaw puzzles and throwing away the boxes so you don't know what they're supposed to look like. And then you're basically trying to like reassemble from scratch. So instead, people have been amplifying just this one gene out, the 16S gene, or sometimes other genes. And here you need some gene that's ubiquitous, so it has to be present in all bugs. It also needs to have some regions that are the same in every bug so that you can use primers to amplify them out. And so my analogy here is that it's sort of like if you had some way of amplifying out all of the corner pieces from all of the different puzzles, then you could line them up and say, okay, these all look the same, so these people all have this bug, or this one's blue, it's probably an ocean puzzle. But some problems are you have poor specificity, so from just that one little corner piece of a puzzle, by analogy, you can't really tell what the whole puzzle looks like. And many puzzles have blue in their lower right-hand corner. So same thing happens here. Most species are not unique when you look at just a little piece of their 16S, like 200 bases or 400 bases. And in addition, so what the problem this causes is that most of the annotations you get back are really only confident at the genus level. You can't go down to species. You also have primer bias. Because you're having to design these probes, DNA probes or primers to amplify out that gene, if it doesn't match the, if you haven't designed a primer to match a particular bug, then it'll just get dropped out completely. And you can also have times when there's some mismatch, and depending on what method you use, you may still see the bug, but its concentration will be much, much reduced in the output relative to what it actually is in the community. So, if primers don't match well, you just won't see the bug. However, I mentioned early in the semester that if you have the box lids, then you can actually figure out the taxonomy really well from the shotgun data. So this works great in well-characterized environments. For example, human gut, we have most of the common members represented with full genomes, at least from some strain, in a reference database. So when you have the box lids, then you can compare each piece to all the boxes and find its closest match, and just tally up how many times a piece hits this puzzle versus that puzzle, and so on. And then you have your species level annotations. The challenge is that there are a lot of full genomes and a lot of potential bugs that could be present in an environment. In the human gut, for example, there are thousands of different species that you might see, and the whole reference database contains tens of thousands of different species or different strains. So, and every genome is unique in a lot of places, although it has other places that are redundant or similar to other bugs. So you need to find the regions of each genome that are good markers. And then the question is, how do you do that? What do you use as a marker for taxonomy so that you're not thinking falsely that there's a particular bug there, when actually the sequence you saw would have matched a bunch of other species? And different methods have taken different approaches to this. I'm going to talk about two of them, Metaflon and Kraken. Metaflon, and there's a Metaflon 2 out now, pre-identify a set of marker genes. So they systematically walk through all of the different species and find out what genes are consistently there within all of the strains in that species and not there in other species, or even genes that are unique to a particular strain within a species. So these are genes that are conserved in a species but not elsewhere. The challenge is when you do this, it could be slow if you used every possible marker from the database. Because you actually have to align your reads with those genes to see if there's a good match. But the nice thing is that they can, most of the time people have really deep shotgun sequencing. For the human gut, you might generate 10 million reads or 20 million reads or 50 million reads. And so it's okay if you just skim off a small subset of genes that are good markers. So they end up using a very small, relatively small database of markers, and then it's actually still pretty fast to search. Kraken is an example of a tool that uses k-mers. K-mer is just a short section of DNA of length k. So you can use an 8-mer, for example. And what they do is they look for k-mers that are unique to one species in the same way as Metaflon, but they store all of the possible k-mers. So in theory, every single k-mer that is unique within a species and doesn't show up in other species is stored in the Kraken database. And then, so it's a very large database, takes up a lot of memory, somewhere in the 50 gigabyte range if you want to use their full database. This would be really slow if you were trying to align to that database, but because we're just matching k-mers from an input read to the k-mer database, it's actually really fast and ultra fast. So Kraken can process a lot more reads, or any k-mer based approach can process a lot more reads per minute than the alignment based approaches. So so that's kind of the trade-off, is that it's ultra fast, but it uses a large database. And what they had to do is subsample down to what they call mini Kraken, which is something like a 4 gigabyte database that you could actually run without a supercomputer. But then you're losing some of the sensitivity, so you won't have, you're not able to use every possible marker as a marker. And they show in their paper that that's not a huge impact on accuracy, but we'll talk about that a little bit more later. And there are some other tools, so you can use Mega Blast to do this search. Fimble is another one that I'm not going to talk about today. So I'm just briefly going to show a couple of results from Metaflon and Kraken. Metaflon uses, so this is actually Metaflon 2, which uses an expanded set of marker genes from Metaflon. If you want to read about all of the nuts and bolts of how Metaflon works, I recommend reading the original Metaflon paper first, and then Metaflon 2 is fairly short and just covers the updates from Metaflon. And one of the nice things about this tool is that it now includes markers for various archaea, various eukaryotes, and also viruses. And here's an example on some simulated data where they're plotting the predicted relative abundance, or the output from Metaflon, and then the expected relative abundance, which is from the simulation. So they know how much of a particular bug they put in when they simulated a community, and then this shows how much is recovered with the different methods. So this is comparing Metaflon metaflon-2, metaflon Mo2, which I forgot to mention, but it is very cool because it can also identify bugs that aren't in the reference database. And then Kraken, which I mentioned earlier. And so what you see is that basically metaflon-2 has very nice correlation with the expected relative abundance and better, lower variance or lower noise than the other methods. Although if you look closely, right along the y-axis, you'll see that there are a number of bugs that are estimated to be there by metaflon or metaflon-2 that are in fact not there at all. So you do get false positives, and occasionally they are relatively high abundance in the estimates. So that's just something to keep in your mind, is that you still can't always trust everything that comes out of these methods. And there's a little bit of work that still needs to be done in the field to control for these sorts of false positives. Now, I mentioned Mo2. So Mo2, you can run, I mean, this sort of test may be a little bit unfair to Mo2 because perhaps it's a little less accurate in the relative abundance, but it also identifies marker genes that, that are conserved in novel bugs that aren't in the reference database. And then it does clustering on those and reports OTUs from the metagenomic data. And so you can actually find metagenomic OTUs, or MOTUs, from the analysis. So it could be that using some combination of the metaflon-2 estimates of the known bugs and then the Mo2 metagenomic OTUs for the rest of the sequences might give you the best features. And Kraken, so Kraken the paper only reports its accuracy at the genus level, which is a little bit worrisome. And I think this is because k-mers are really tricky to use as classifiers. Like, you might see a sequence that has a bunch of k-mers that seem to uniquely match one species, and then all of a sudden it switches over to a bunch of k-mers that seem to uniquely match another species. And we don't know if this is because the read is chimeric or the database isn't complete, and actually all those k-mers are present in both of the species. And it's actually very tricky to know how to do the voting. So if you see like 30%, here's an input sequence, you take every sequential k-mer and match it to the unique markers. If 30% match one bug and 35% match another bug, and the other 35% are matching a whole bunch of other random bugs, should you call the one with a plurality or should you toss it? And that voting approach hasn't really been explored yet. Definitely an area of future work. You can see here that Kraken gets very high precision, comparable to some of these other methods, but is much, much faster. So this is the plot on the right is showing how many reads per minute it can process. I think as long as it's not taking days to process your sample, you're probably more concerned about the accuracy of your results, because the processing time is fairly short relative to how long it takes to collect the samples and do all the extraction and sequencing and so on. Also, as I mentioned, Kraken has very large memory requirements. You can use mini Kraken, it's not as comprehensive. And they don't really assess the accuracy in a thorough way of the relative abundance estimation. So it can tell you if a genus is present, but not necessarily with accuracy what the abundance is. And this could be a little bit of a problem. I mean, it depends what sorts of tests you want to run. But so I personally am a little concerned about the lack of specificity at going down to species level, and the lack of validation on the relative abundance information. On the other hand, it's super fast and very comprehensive, so it might work better in more shallow shotgun data than the other methods. So in summary, shotgun metagenomics can be used for identifying species. It's far superior to 16S in this respect. All of the methods that try to annotate species or strains out of the shotgun data have the same goal. So they want to identify reads in the input data that uniquely identify a particular species. So Metaflon 2 is very comprehensive. It's reasonably fast. The new version covers eukaryotes, viruses, strains, and so that's a very nice improvement. And you get pretty good estimation of the relative abundances with a few false positives. Kraken is extremely fast. It's able to use, if you have the high memory version, if you can use the high memory version, it's able to use all of the input reads that could possibly uniquely identify something. So if you have shallow data, it might be good for that. Doesn't seem to be so good at species level, and the relative abundance quality is unknown. And that's all for this video.